paper 2. Thank you so much for joining. We are not going to waste too much time. Let's start without wasting time to check and see all the topics in details. How can we manage to get a distinction? I know those people who joined us with the paper 1. I hope you guys you achieved it. So let's go for paper 2 and then find out what we can do for paper 2. All right. The first question. They are saying that describe the structure of DNA. When the question comes like this, what do you say? When you're talking about this structure of this, describe the structure of DNA, automatically you have to describe as DNA is double helix, weak hydrogen, it has weak hydrogen bonds. These hydrogen bonds, they join together, the two strands to form a double helix. You describe the real structure, what you see on DNA. For example, when you look at it, when you look at the structure of DNA, you describe what you see there. DNA is made up of nucleotides. The nucleotides are made up of phosphate, sugar, oxidable sugar, and then a nitrogenous base. A nitrogenous base are linked together by weak hydrogen bonds. So you explain what is it. So we go the next question. So this one is 100% done. Okay. Sorry. Yeah, so this one is done. When they say what are the monomers of DNA, here we talk about the nucleotides. The nucleotides are the monomers of DNA. Nucleotides. Who discovered the structure of DNA? Here you talk about, because they can ask you a question that who discovered the structure of DNA using X-ray diffraction pictures. Who discovered the structure of DNA? James Watson. Who were involved in the discovery of DNA? James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. So who discovered the structure of DNA using the X-ray diffraction picture. So when you talk about the discovery of DNA, we are not too much asking you a lot of story, what was there and before and after. No, we just want to know who discovered the structure of DNA. So in this case, who discovered the structure of DNA using X-ray diffraction pictures? Here you talk about Maurice Wilkins and Rosalind Franklin. So these are the two people who were who used the X-ray diffraction pictures to identify or to discover the structure. In which year was the Nobel Prize given for the DNA? Here the year in which the DNA Nobel Prize was given, it was 1962. Yes. So other than the nuclear, okay, now we are starting. Other than the nuclear DNA, yes, other than the nuclear DNA, which other types of DNA do you know? We have the mitochondrial DNA, and then the chloroplastic DNA. So you need to know these types of DNA or where they are located. Where they are located will determine other types of DNA. The next question is, what is DNA replication? Is the process by which DNA makes exact copies of itself. When does it occur? It occurs when does it occur? This one is occur, won't come. When does it occur? It occurs when exactly does it happen? It occurs during interphase. Where does it occur? It occurs, where does it occur? Here it occurs in the nucleus. And why does it occur? When you talk about why does it occur, you need to know all these answers. Yes, why does it occur? It occurs so that it increases the chromosome number to double the genetic material, so that the new cell which is coming in, when it divides equally, so that there is no loss of what? Genetic material. So it doubles, it helps in the doubling of the chromosome number or the genetic material. Uh-huh. We got the next question. They are saying that how does it occur? Here you explain the process of DNA replication. When you talk about the process of DNA replication, DNA double helix unwind, weak hydrogen bonds break, they form two separate strands. Each is acting as a template formation of messenger. So you explain DNA double helix, DNA double helix unwind, DNA double helix. That's a mark. Weak hydrogen bonds break. Yes, to form two separate. This word is very, very, very crucial. Some people they forget about it. To form two separate strands, each is acting as a template. Using free floating DNA nucleotide. So remember, in the nucleus we have the DNA nucleotide and RNA nucleotide. So because it's DNA replication, we want to see that you're using free floating DNA nucleotide. From the nucleoplasm, a complementary DNA strand is formed whereby A pairs with T and then G pairs with C. Some people they say that where A goes with T, in most cases sometimes we don't give you a mark when you just write like that. Where A goes with T and then G goes with C pairs with or is complementary to. Yes. Two new DNA which are formed which are genetically identical. They are not just two new DNA. They are genetically identical, each with one original strand and one new strand. So this process, you don't know it, take a screenshot of it so that you're able to remember and give a tick of it whenever you are done. Uh-huh. Um, We continue to the next question. They are saying that how does it occur? It means that you are describing the process of DNA replication. All right, we got the next question. They're saying that what are some of the functions of DNA profiling? Here you talk about some functions of DNA profiling. When they bring a question about DNA profiling, in most cases, we bring a scenario, something like this, and then we ask you some few questions about it. So you have to know at least when they bring. Let me explain. For example, if they bring a question and this question is about criminality, make sure that uh, they ask other than that, what is another function of DNA profiling? So it means that you don't talk about the criminality. If it's about the paternity test, then it means that you don't talk about that, you only talk about something else. So you need to know that don't just give the answer because DNA profiling is simple and easy for you to understand. So make sure that at least you eliminate what they have asked you. Mhm. Then when they ask you the advantage, disadvantages, or uh, the doubts, or debates about DNA profiling, you can talk about is expensive. You can talk about. They can use it to frame someone, even if you are not the one who did for example, the crime. They can use to find someone. Yes. Someone is saying can't hear anything. Okay. Am I too fast? Am I too fast? Can I reduce the speed, Nyana? All right. So we are saying that. So from here, DNA profiling, you need to know to know how to do the calculations for bases. Remember, when you do base calculation, for example, they can say that 20% is cytosine. What is the percentage of adenine to guanine? What is the ratio of adenine to guanine? So I will know that cytosine, what they gave you is what you start with. So C goes with G. And then if C is 20, G is also 20. Because this is percentage, then I'm gonna say that 20 plus 20 is equal to 40. So it means that the thymine and adenine the balance will be from 100 minus 40 here, which gives me 60. The 60 will be for 2. So it's 60 divided by 2, which gives me 30. So it means that this is going to be 30. So also this is going to be 30. So but what are they looking for? They're looking for adenine versus guanine. So I'll come back and ask myself, what is guanine? Guanine here, guanine here. We see it is 20. Adenine here is 30. Then I make it the simplest ratio, and then I'm able to divide by 10, divide by 10, so it becomes 3 to 2. So that is the ratio. They're saying that adenine to guanine. So that is the ratio. And guess what? When they're bringing in the questions, yes, when they're bringing the question, they can start with the 2 to 3. So because when you are rushing, you end up getting it lost. And then number 2, sometimes we don't bring it in terms of what's happening. All right. Let me see, why is it doing that? Let me see why it's doing that. System error. Guys, sorry. I want to see why is it doing that. Okay. Still system error. Okay, why is this doing system? All right, let me check. I'm checking here to find out. Okay, let me access. Then, yeah, I think is back. This is SE. Yes, I think now it's fine. Sorry for that, guys. We continue. They can also give you a question whereby they're asking you that if DNA has 800 bases, it's not percentage, and the 200 is adenine. So what is the percentage of cytosine? So they want the cytosine percentage, of which I have to do the same thing that adenine. What they give you first is what you write. Adenine goes with thymine, of which this is 200, and then this is 200. So when I combine, 200, also this is 200. When I combine, they become 200 plus 200, we get 400, isn't it? So the balance of cytosine to guanine goes as 800 minus 400, which gives me 400. So divide by 2 so that I'm able to get 200 here, also 200 here. So it means that the percentage, so this is 200. So but they want the what? The percentage. So what am I going to do? I'm going to say that 200, since I got 200 out of 800 times 100, convert it into percentage, and then now I'm able to get the percentage as, what is the percentage? That is 1 out of 4 times 100, which gives me 25%. So that is how we do the calculations. Do you understand? Yes. So now we go to the next question. So there's a base calculation. Please make sure that at least you know, so that you don't lose those 2 marks to huh. Then they're asking a question. So this is done. What is the function of a messenger RNA? The function of messenger RNA is to carry coded information. It's to carry the coded information for protein synthesis. That's the major function of the messenger RNA, is to carry the coded information from DNA for protein synthesis. So if they ask you that, and why is this messenger supposed to have that specific sequence? So it's basically it has specific sequence because it carries that specific information, which is supposed to determine the specific sequence of transfer RNA with the amino acids leading to a specific protein. Yes. So it basically, the function of the sequence is basically that sequence has a meaning, and that meaning can be transcribed by the transfer RNA coming in that specific sequence with the amino acid. And after the amino acid join together, they're gonna form a specific protein. So that's what it means when they say that. Why should it have specific sequence? What is protein synthesis? Protein synthesis, the process by which proteins are being what? Made. Yes, in the cell. Mhm. So that is it. In which organelle does protein synthesis? In which organelle do we have transcription? Here we're talking about the nucleus. And on which organelle do we have translation? Here's translation. The organelle is ribosome. The ribosome. They are saying describe the process of transcription. Here you talk about the process of transcription. How do you talk about the process of transcription? You're talking about DNA double helix, unwind, weak hydrogen bonds, break to form two separate strands. To form what? Two separate strands. One is acting as a template for formation of messenger using free floating DNA. Sorry, using free floating messenger RNA nucleotide from the nucleoplasm. So you explain the process of trans. At this time, you have to be knowing that. If you don't know it, take a screenshot. Yes. Take a screenshot, DNA double helix unwind from unwind, we hydrogen bonds break before they break. Once they break, uh, meaning that they unzip. One strand, or single strand will act as a template for formation of a what? A new strand using the free floating messenger RNA nucleotide. You understand? Remember there, it was using free floating DNA nucleotide. So make sure that at least you know that. If it is complicated for you, you will have to memorize it. At least memorize, write, pass, forget. Yeah, at least when you have it. Uh-huh. Any question, guys? Any question from the? I don't see any question from the chat. And I don't see any question from the chat. But if you have any question, let me know so that you're able to solve it out. Or if you don't understand something, any concept, don't forget this is a live. Some people, I like these people when they always tell me, it's pre-recorded, pre-recorded. All right. It's not pre-recorded. All right. We are saying that describe the process of translation. Here now you describe the process of translation, whereby you tell us that according to the messenger RNA, codon, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acid are joined together by peptide bond to form a required protein. So that's what is supposed to be written when it comes to that question. You have to do that. Take a screenshot if you don't understand it or if you don't get it right. So that you are able to memorize it and get it on that actual time. Let me know which school are you coming from, which area are you coming from. Yeah, I want to know who are those people who are with me. Yeah, so that yeah. We got the next question. We got the next question. We got the next question. The next question is saying that what is the difference between the DNA nucleotide and RNA nucleotide? When you talk about the DNA nucleotide, we are looking at the structure of DNA nucleotide. Remember, we said that the monomers from our question above, we said that the monomers of DNA, what are the monomers of DNA? We said it's a nucleotide. If the nucleotide so is made up of phosphate group, deoxyribose sugar, and then the nitrogenous base. So that is the nucleotide. So if they say what is the difference between the nucleotide of DNA and that of RNA, they are not looking at the strand. No, they are looking at just the nucleotide. So in this case, you talk about DNA has a DNA is deoxy, has a D. Let me first draw it. You look at, since the phosphate group is the same, so you only talk about DNA has deoxyribose sugar, RNA has ribose sugar. While DNA has a thymine, RNA has uracil. That is it. Then they ask a question. What are the differences between DNA replication and transcription? When you talk about DNA replication, what does it mean? DNA replication. What does it mean? All right. In this case, we look at what happens during DNA replication. Where does it occur? How does it basically? DNA replication, two strand act as a template. Well, transcription, one strand act as a template. In DNA replication, you form two genetically identical DNA molecule. Transcription, you form messenger RNA. In DNA replication, we use free-floating DNA nucleotide. In transcription, use free-floating RNA nucleotide. So you give the differences between what happens in DNA replication, which does not happen in RNA. You understand? In DNA replication, we're gonna have thymine. In RNA, in transcription, you're gonna have uracil. So you see that, yeah, you see that you are able to write the differences which happens in DNA but does not happen in RNA. All right. We go to the next question. Now let's try to answer these questions. So you have W. Which molecule is it? Which molecule is it? W. Which molecule is it? W. Which molecule is W? All right. Which molecule is W is nucleotide. Remember, we said that nucleotide is the one which makes up the backbone of the DNA. So it's a nucleotide. What is V? We say that this is thymine, because this is thymine, and then this is what? If this is thymine, this is thymine, so automatically then this is gonna be adenine, isn't it? And then what is U? U is the DNA molecule. What is Z? Z is weak hydrogen bonds. So now we come back here. The molecule W, we know. We said is what is molecule W? Molecule W, we said is nucleotide. What is U? U is DNA molecule. Part W, which molecule W labeled X and Y. X is phosphate group, and then Y is oxyribose sugar. Bond Z is weak hydrogen bonds, and then nitrogenous base V. Nitrogenous base V. Nitrogenous base V. We say it is adenine because this one is thymine. So you have to look at the how it is being asked. Where in the cell does this process take place? It takes place where? In the nucleus. It takes place in the nucleus. nucleus. Name the process of cell cycle where replication takes place. We said it takes place in the inter inter interface. So that's what it means, and that's what you're supposed to know when it comes to that. Then they are saying that the diagram below shows the part of messenger RNA molecule. Part of messenger RNA molecule. So let's find out how many codons. Remember, you have codons, and anti-codons. This is for DNA. This is for RNA, messenger. Then this is transfer RNA. So whenever they say codon, it means that this is a messenger RNA. Even if I don't read what is written up there. So how many codons show in the diagram? It's 1, it's 2, it's 3. So that is done. They're saying write down the complementary base sequence of DNA strand from one of the messenger RNA. They said what formed one. Okay, so let me first clear up and see. They're saying that they said write the complementary base sequence of DNA. So it means that you have to convert this into DNA. So this is gonna be A T G, because in DNA A goes with T, while in RNA A goes with U. So done with that. Explain the purpose of specific sequence of codon in messenger RNA molecule. Specific sequence of codon in the messenger RNA molecule. So I come back here and then I look for it. What did we say? Actually, this question is similar to the question we have just answered here. So what is the specific sequence? What is it for? Basically, this specific sequence explain the purpose of specific sequence of codons in messenger RNA. Remember, messenger RNA carries a specific information, coded information from DNA. So that sequence will determine the sequence of amino acids brought by a transfer RNA during translation, and that will lead to a specific protein. So it will determine the sequence of amino acid, or it will determine the sequence of transfer RNA with their amino acids. You understand? So leading to a specific protein. So that's what you need to look at. tRNA molecule carries complementary bases for a particular protein, for a particular codon. tRNA molecule carries complementary bases for a particular codon, Okay, write down the complementary base sequence of tRNA for codon 1 messenger shown in the diagram above. So it means that they want codon 1 but for tRNA. If this is messenger RNA, they want now the tRNA. So automatically I'm gonna say that U goes with A, and then A goes with U, and then G goes with the G. So basically it's gonna become like that. We got the next question. They are saying that briefly describe the role of transfer RNA in the translation process. RNA, the function of RNA, it carries specific amino acid to the ribosome when its anticodon matches with the codon of messenger. It is here. Let me show you. It is here. Each transfer RNA carries a specific amino acid when the anticodon, on tRNA matches with the codon on messenger RNA. You see? So basically that is it. So the required amino acid will be brought. So when it carries an amino acid to the ribosome, when the codon of messenger matches with the codon, the anticodon of transfer RNA. Yes. Take a screenshot from our distinction material book. Those people who don't have distinction material book, you know where to access it. You just click on join and then you're able to access that book for distinction material book. All right. We continue to the next question. We continue to the next question. All right. Let's label this. What is this? That is 5. It is the amino acid. What is this molecules? Sometimes they draw it like this. Sometimes they draw it like that. So this molecule is what? Is ribosome. And then what is 7? 7 is 7 is codons, because this is a messenger RNA. Messenger RNA. So it's a codon. And then what is 1? 1 is a DNA molecule. What is 2? 2 is a template, because this one is a messenger and is facing this side. So it means that this messenger was formed from this. So it means that 2 is a template, and then this is just a DNA strand. Okay, let's find out. Turn out stories. All right, let's find out. Let's find out and then see. Provide the labels for molecule 1. So we saw molecule 1 is where's molecule 1? In a molecule, organelle 6 is organelle 6. We say that is ribosome. Give the number of the part that represents DNA template. DNA template strand. DNA template strand. Give DNA template strand, it is number 2. So because we said this one is facing this side, which is messenger, therefore automatically this is DNA template. Then they say that monomer of proteins. Monomer of proteins, these are amino acids. Whenever they say monomers of proteins, these are amino acids. So automatically the monomer of protein in that case, sorry guys, yeah, the monomer of protein in that case is gonna be number 5. So these are amino acids. This is not yet a polypeptide chain. Why? Because they are not joined together by peptide bond. You see, they are still free. So automatically they are not polypeptide chain. The thing, describe the translation as occurs on organelle 6. So you describe the process of translation whereby according to DNA, according to the codon of the messenger, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acids are joined together by peptide bond to form a required protein. Yes. The way. Are we there? Are we there? Are we in class? Are you getting it? Are you getting it? Are you getting it right? Yes. If you guys have a question, you let me know. Irene, are you in class? All right. We continue with go the next question. They're saying that codons. State the two difference between DNA. Did I skip? DNA, provide a DNA sequence for glycine. With glycine, we come here, they're saying DNA sequence. So let me first clear up. Glycine is this, ne? But they want DNA. Automatically, this is tRNA, this is messenger RNA. So I have to be with DNA down. So it means that it's gonna be C, T. So that will be the answer for that case. And Sanele from KZN. Zululona. All right. Thank you so much for joining. All right. We continue to the next question. We are saying that codon for proline. Codon. Remember, codon is, it means that it is the messenger. We say that you have code, you have anticodon, we have codes. So in this case, codon means messenger. So messenger for proline. So it means that they are looking for this. So it's gonna be CCU. So when they ask you a question, when they ask you a question, and that question is DNA codes, DNA codes, and they're asking you what will happen afterwards. For example, they're asking for tRNA. So you have to make sure that at least tRNA, you convert it into messenger RNA, then you take it to tRNA. You don't just say it's from DNA and direct tRNA. No, you have to first convert it into messenger, and then after that, you convert it into tRNA. All right. So we continue. We continue to the next question. We continue to the next question. State the difference between the nucleotide of DNA and the nucleotide of RNA. So here we talked about them, and I say that make sure that you you talk about it, and then you you only focus. You don't talk about the whole structure of DNA. You only focus on the nucleotide. This one is, we say that is the same in both. We say this is the deoxyribose sugar in DNA, and then for the case of RNA is ribose. And then we say this is thymine in DNA. For the case of RNA is uracil. So that is the difference they are looking for. Before we go to the mutation, do for me a small recap. Let's recap together, me and you, before we go to the mutation. Remind me now, what is W? W means DNA molecule. What is X? X means messenger RNA. What is Z? Z means Z means Z means a ribosome. This is the ribosome. Sometimes it comes as one, sometimes it comes as two section, a major subunit and a minor subunit. So this is the ribosome. So this is what? This is amino acids. These are amino acids. Those are amino acids. What is this? This is, what did I say? I didn't say, did I say amino acids? No, no, no. This is transfer RNA. Transfer RNA. These are amino acids. What's happening? This system. Okay. So that is it. So we are saying that this is amino acid. These are transfer RNA. This is a transfer RNA also with an amino acid on top. So this is a peptide chain. It's not a protein yet because it becomes a protein if there are 50 amino acids. What's happening? It only becomes a protein if it is 50 amino acids. If it is not that, it is not yet a protein. We got the next question. What are they saying? What is Z? We said Z. Z is ribose, sugar, ribosome. Something is not. Don't worry. Yes, we have just started, ne? Or peel. We just started. Let me check what is the problem with this. Why is it making noise? Okay. So we are saying that what is X? It is messenger RNA. And what is this? What is this? We say that it is tRNA. What is this? This is amino acid. What are the monomers of proteins here? It is the amino acid. How do we call the bond between the two amino acids? Between these two amino acids, we said we call the bond peptide bond. And how do you call the chain? We call it polypeptide chain. How do you call this? Call it the nuclear membrane. And how do you call the hole between the nuclear membrane? So we call it a what? Nuclear hole. We got the next question now. Got the next question. Thank you so much. Paper one was good for me. You helped. Yeah, let's focus on paper two now. But Piro, let's focus on paper two so that we also make it and then we smash the paper. We'll smash it. Don't stress. We'll smash it. And then we get good distinctions. All right. We continue. A mutation has occurred on a section of messenger RNA molecule as shown below. Right. Name the type of mutation that occurred. So which one occurred? So this one is fine. Check here and check here. Fine. It's 1, 2, 3, 4. So the fourth triplet, the second base was of C was replaced with here. This one now was replaced with U. So that's the meaning. That is the meaning. So name the type of mutation shown in the. So how do you call the mutation shown here? How do you call this? Because it's just a mutation. So we call it gene mutation. Gene mutation or point mutation. So we are going to the next one. We're saying the table below shows some of codon and amino acids that they code for. Amino acid that they code for. So in this case, what are they saying? The table below shows some of amino acid they code for. All right. Oh, they are saying that give a reason for your answer. Why do I say this is a gene mutation? Because the second trip, 1, 2, 3, 4. The fourth triplet base, C was replaced with U. That's what you're supposed to talk about when they ask you about. You have to tell us which triplet changed and what changed from what to what. Then we got the table below shows the messenger RNA codon and amino acid that they code for. State the number of different amino acids coded for by the original sequence of messenger RNA molecule given above. They want to know the number. And the key point here, different. They're not saying how many of them. They want to know how many are different. How many are different? We go back and see how many are different, because we have to check and see, do they code for the same information? If they don't code for the same information, what are they coding? What do they code for? So here I will see that this that code for AGAGU AUG AUG codes for methionine. GAA codes for glutamic acid. AUA codes for isoleucine. Then CCG codes for CCG proline. CCA codes for proline. So it means that now this is 1. We count it as 1. GGA glycine. So it means that it's gonna be 1, 2, 3. This is counted as 1, 4, and then this is 5. So there are 5 of them. Yes, if you check there are 6, but because one code is for the same thing, because of the word different, unless it was not different types. But if it's the different types, then automatically the one which is the same, we don't repeat it. So there are 5 amino acids. Give the anticodon of tRNA molecule that carries the amino acid isoleucine. They are saying they give the anticodon on tRNA molecule that carries for isoleucine. Isoleucine, it's here. But remember, this is messenger RNA. So they want it to be written as tRNA. So it's AUA. So convert it into tRNA. So it's gonna be U. It's gonna be UAU. So that will be the answer. I hope guys we are getting it right. Because we are going together, né? They're saying that use the information in the table below. Describe the effect of mutation on the protein. Oh, I need to know which type of mutation is this. So what I'm going to do is I need to find out. The first thing you need to do, guys, né, is this. This is what you need to do. So identify where the problem happened. It happened there. So identify what is this. Here they said is messenger. And what is in the table? It's also messenger. So do I have to change these codes? No. If this is different, for example, if this is messenger RNA and then this is transfer RNA, then you have to change the what? The sequence. Whereby you're gonna tell me that C goes with G, C goes with C, and then G goes with the C. But now since this is messenger and this is messenger, I'm gonna have to read it the way it is. So CCG will code for what? CCG codes for proline. Proline. And then this one, CUG codes for what? CUG, it codes for leucine. Okay. So you see that? You tell us now, will the protein change? Yes, the protein will change. How will the protein change? Sorry. How will the protein change? Someone is asking me a question I don't understand. Please stop showing the answers starting from 21. What's that? Starting from 21. What is 21? I don't understand. But what you're saying, I hope you are getting what I'm saying. I don't know whether you want to write down or what. So you're saying that how is the mutation? Describe the mutation. How am I going to explain the mutation? So we have seen it that this, and this is the same. So it forms proline. And then this one forms leucine. Will the protein change? Yes, the protein will change because you have formed different amino acids. Unless if this one was also proline, then the protein wouldn't have changed. So now how do I write? You tell us that. You come back here and then find that. The 1, 2, 3, 4. The fourth triplet, C was replaced with U. Okay, that's a mark. Whereby CCG codes for proline and CUG codes for leucine. This will result in a different protein. So that's what it means. That you tell us that which code, which triplet was affected, and when it was affected, what was being coded for before it changed, and what is being coded for after changing. And then after that, you tell us that will the protein change or will the protein not change. So you have to compare the two and tell us what is really happening. So we go to the next question. I'm going to give you 5 minutes. 5 minutes, you guys. 5 minutes as a break so that you can give me the answers of this. So when I come back, I'll be able to shoot from meiosis and then I continue. So 5 minutes break. All right. Okay, guys, I'm back. uh I see people. Yes, Dr. John Dobe, high school. Oh, Sifiso, Kwamashu, ne? Ah, that's good. That's good if you're joining. See people saying that genetic variation that cause offsprings to be better adapted into a particular environment to ensure survival, right? In paper one, I was cooked. I'm writing paper two. Wednesday economics. Don't worry, we pray for you guys so that you pass and everything becomes good for you guys. Ne? Yeah, we are praying for you for a better future. Better future. All right, guys. Let's start. Let's start, and then we continue from where we stopped, so that we see that we get these things sorted out by the end of the business day today or tonight. All right. Uh-huh. Uh, they are saying that name two importance of meiosis. When they talk about importance of meiosis, what is meiosis used for? What is it for? Meiosis is responsible for gamete formation. Gamete formation. Actually, it results in the formation of haploid gametes. Haploid, haploid gametes. Let me show you. Oh, someone said, show me the answers. Ish. It results in the formation of haploid gametes. That's number one. Number two, meiosis. Hey, I've gone so far. What's our distinction? What you book? Wait. Oh, oh, it's far, it's far, it's far. It's far. It's here. No, man. No, man. No, man. No, man. Where did I write it? Okay, let me just tell you. Yeah, it's here. Eh, Formation of haploid gametes. That's the first point. Formation of haploid gametes. Number two, maintains the chromosome number from generation to generation. Maintains the chromosome number, constant chromosome number. Why? Because remember, you are 46, the mother is 46. So meiosis result in the half the number. So you're gonna be 23, the sperm, and the ovum is gonna be 23. So that when they combine again, you go back to 46. So it brings about the halving effect, which solves the problem of the doubling effect. So that's why you say that it maintains the chromosome number from generation to generation. The third one, it brings about genetic variation. Please don't say it brings about variation, because variation can be genetical or it can be non-genetical. So it brings about genetic variation. All right. We continue to the next point. Sifiso, are we together? Are we together? Are we together? Uh-huh. You know, Sifiso, you are my boy, ne? You know that. I want distinctions. All right. Uh-huh. We continue to the next question. We continue to the next question. Uh, which structure does meiosis take place in plants? In plants and animals. Remember, plants, there is males and then there is females. And animals, there is males, also there is females. So in animal, the female, it occurs in the ovary, produce of ovum. In males, it occurs in the testes to produce sperms. In females, it occurs in the ovary, for the case of plants, to produce ovules. In males, it occurs in the anthers to produce what? To produce pollen grains. So that's what it is. You understand? Yes. So automatically, this is what they're talking about when they ask you all these processes. Where does it occur? If you're using a distinction material book, yes, you know what I'm talking about here. This is what I'm talking about. You can take a screenshot if you are interested or you don't have it in somewhere. We go to the next question. Describe the structure of a karyotype. How am I going to describe the structure of a karyotype? So basically, karyotype, you describe something like this. Karyotype is made up of 46 chromosomes. You talk about 46 chromosomes, a human karyotype. Yes. 46 chromosomes. From chromosome number 1 to chromosome number 22. Yes, those pairs, we call them orosomes. The 23rd pair, we call it gonosome. These pairs are arranged, they're of different sizes. You see, they're of different. They're arranged, those which are of the same size, they're arranged in the same what? In the same size. The 23rd pair is made up X and Y. Yes, X and Y, whereby if X and X meet, you're gonna form a female. And if X and Y meet, they're gonna form a male. X is longer than Y. So you describe the karyotype. So they can bring this question and then it's 6 marks. All right. So we're done with that question. So we go to the next question. Describe the process of crossing over. So those people about the karyotype, if you are looking for the karyotype, this is what they are looking for. Human karyotype, 46 chromosomes, arranged in pairs in different forms, the size and, you understand, in the composition from 1 to 22 orosomes. 23rd is gonosome, made up of X and Y. And then if X and X is long, so that is what you're supposed to explain. Then the question is saying that describe the process of crossing over. So you describe the process of crossing over based on this. Say that homologous chromosomes align together. They align together. All right. One chromatid of each chromosome overlap with chromatid of its homologous partner, which we call adjacent. Adjacent. Adjacent chromatid of each chromosome or of each homologous chromosome overlap. Meaning that, remember, you have this chromatid and you have this chromatid. But the one which are adjacent one another is the one which overlap. That's why you want now to add the word adjacent chromatid of homologous chromosome, yes, of each other, adjacent to each other, so they overlap at a point called chiasm. You understand? Then crossover takes place at a point called chiasm. The chromosomes separate in such a way that each has new genetic material from both chromosome that is shared, meaning that each will have to share the content from the father and the content from the mother, like that. Remember, one chromosome is from the father, one chromosome is from the mother. So please make sure that you know how to operate the non-disjunction. All right. We continue in saying that what is the importance of crossing over? This one brings about genetic variation. Not variation, I said genetic variation. They're saying that other than crossing over, which other cause of variation in meiosis? The key point is there in meiosis, whereby in here you talk about, what do you talk about here? Here you talk about uh, random arrangement of chromosome during metaphase, and then you can also talk about mutation, because mutation could be occurring in meiosis, and random arrangement of chromosome during metaphase. That is the major one. Then we go to the next question. Give two differences between meiosis 1 and meiosis 2. In this case, it's easy. It is easy. You look at phase by phase. You look at phase by phase. In meiosis 1, you talk about chromosomes arrange themselves at the equator in homologous pair. When this is mera phase, you see, while here chromosomes are line up at the equator individually, and then whole chromosome moved opposite poles. This is the anaphase. Daughter's chromosome or chromatids moved opposite poles. Still is anaphase 2. Two cells at the end of this division. This is telophase 1. And then 4 cells are formed. This is telophase 2. They're saying that chromosomes number is halved. Still, this is telophase. Telophase 1, because we have what called a cytokinesis and karyokinesis. Cytokinesis means cytokinesis means division of the nucleus. Karyokinesis is cytokinesis, the division of the cytoplasm, while karyokinesis, the division of the nucleus. So when there is karyokinesis, the chromosome number is being halved. Then there is crossing over takes place. This is prophase, and then crossing over does not take place. So this is prophase 1 and prophase 2. There is no such. So you have to know how to differentiate between the two. So if you forget, look at least give 2 of them, because the question is asking you here, give two differences. So it means that you're supposed to know that. And then they're asking you give the differences. You tabulate the difference between meiosis and mitosis. Here you come back here and you look at it, and then you say that the differences here in mitosis, you will see that it occurs in the body cells. It occurs in all body cells. Or here it occurs in reproductive organs only. That's where we ask you that where does meiosis take place? It occurs in the gametes, in the reproductive cells. Production of the cells you form, they are genetically identical. All these ones, they are genetically non-identical. They are not identical at all. That's why your brother, you don't resemble your brother. If the so-called even the so-called identical twins, to me, they are not identical. Because if you check it properly, there is nothing identical there. You see, this one is big, this one is small, this one is tall, this one is short, this one is light skin, this is dark skin. However much they look identical. Even when you check, even this one is a little bit clever, this one is a little bit not that as clever as one. So there is differences there. You understand? Even if it's identical twins, they'll have different mood swings. They have different moods. So it means that they might look different, might look the same outside, but inside totally different. And what causes that? It's because of the meiosis. So we call it the variation. They are genetic. Yes, they look to be the same. They have characteristics which they are same, but there is some differences which are there. So result in 2 daughter cells, and then here it result in 4 daughter cells. So you at least memorize 2 of them. Memorize 2 of them. Then we go the next question. They are saying that let's go the questions and find out. What is A? We say that we are saying that A is spindle fibers. State 2 characteristics of homologous chromosomes. Here we have 2 characteristics of homologous chromosomes. We say that they have the same size, then they have the same shape, same shape, and then you find out that they have the same location of genes. Same location, not same genes. The genes are located in the same spots. How many each of the following present in the diagram? Chromatids, it's 1, 2, 3, 4, 5, 6, 7, 8. Centromere. It's 1, 2, 3, 4. They're saying draw the cell or to show one of the cells that would form at the end of telophase. So it means that this one is gonna go one side, or this one also gonna go another side. So if I draw a cell here, you have to show all the, oh, you need to draw it properly. Use this one. You need to draw it properly. And then this one is like that, and then this is shaded. And then this one like that, and this is shaded. And then the other one, this one, will also be the same. And then, but this one is on the bottom. This is the bottom. And then, yeah, you shade. And then after that, they said at the end of telophase, so then maybe it has the nucleus, and then you put the what cell membrane, nuclear membrane, then you label the chromosome, and then you put the caption. So if you don't do that, these variations, we need them. We need them, we need to see them, make sure that they are there. We go to the next question. All the way to, are you there? I'm seeing you here. They're saying that, what is A? A is the cell membrane. What is B? It says a chromatid network, a chromatid. And then what is C is the spindle fiber. What is D is the centriole. And then, yeah, this, which phase is this? Is the metaphase 1. This is the, did I say metaphase 1? Yes. All this one is, they are separating. It's anaphase 1. And then here we see it is the telophase 1. So that is it. Identify the phase represent diagram 1. We said 1 is metaphase 1. Diagram 3, it is the telophase 1. Give the letter of the part that contains DNA. It is B, because this is a chromosome. So it's B is a chromatid or chromosome. This, attaches itself to the centromere. Centromere. It is C, which is the spindle fibers. Then they're talking about forms the spindle fibers. From the spindle fibers, it's the centrioles. It's the centrioles which form the spindle fibers. Then they are saying that, where am I? Okay, saying that name the organ in human where meiosis takes place. We say that it occurs in the new what? Where meiosis takes place, it occurs in the male. So it occurs in the testis. Testis. That is where it occurs. Then we continue to the next question. How many? They're saying that this is a karyotype of someone having Down syndrome. Down syndrome. Okay. Saying that label A, it's homologous chromosomes. How many autosomes are in this nucleus? Remember, autosomes is from pair number 1 up to pair number 22. So we need to find out. So this is 12. This is also another 12. This is also another 12. So that is 36. Then 7, 38, 39, 40, 41, 42, 43. 44, 45. So they are 45. Why? Because the 21 is supposed to be 44, but because of Down syndrome, plus 1 chromosome, which gives me 45. 45. So there are 45 chromosomes. 45 autosomes. Name the type of chromosome at point this 23. Name the type of chromosome. This one. Let me clear up. How do you call this? We call him gono. gonozome. Then they are saying that what evidence suggests that this karyotype is a male? Because the 23rd pair has X and Y chromosome. Name the type of mutation represented in the diagram. This is called the chromosomal aberration or chromosomal mutation. That's what you are trying to say. Sir, I really love the way you thank you, Dimpako. DNA profiling, we finished it, you guys. Yeah. And I really love the way you guys study with me. Yes, I really love it because it doesn't make sense if you are just online alone and then you're just talking to yourself. Mm. Uh-huh. We continue. Describe what is Down syndrome. What is Down syndrome? Describe the process of Down syndrome. Here you explain how Down syndrome forms. And remember, it's as a result of non-disjunction. Non-disjunction means failure of the chromosome to separate during anaphase. You understand? So, basically, Down syndrome, when there is non-disjunction, we see that you're supposed to have, let me do like this. You're supposed to have a cell with the two chromosomes, with the four chromosomes. Let me just do them like this. And then during the normal reaction, you expect it to have to separate properly. You understand? Separate properly. So this is anaphase, this is metaphase, and this is anaphase. So that it results in this and that, and then this and that. You see? So it means that when it separates, one cell goes there and then another cell goes there with the two of them. But when non-disjunction occurs, so what happens here, the whole set will go to one side. So I'm going to have this, then this one is going to separate properly. And what causes that is because of the failure of the spindle fiber to form. So basically now, what happens here? These two, they shift. These two, they go to one side. But remember here, they were separated, whereby this one goes one cell, another one cell. So now, it means that if this is 23, it's supposed to be 23, 23. So now, automatically, because the whole thing, the chromosome number 21, the whole set has gone to one side, here. You see? So it instead of 23, it's going to have 23 plus this extra plus 1. So which gives me 24. And then this one, this side, this cell, supposed to be 23. So it's going to be short of one chromosome because it went this side. Therefore, it's going to be 22. Now, so if, remember a normal gamete, now if this is a male, and then now during fertilization, you're going to have the female. And the female, nothing happened. So it means that the female is going to bring 23. So if this 23 of the female fertilizes the 24 of the male, so it's going to become 23 plus 24, which gives me 47. And the normal cell is supposed, normal fertilization, if male and female, is supposed to be 46. So automatically, the individual is going to have 47 instead of 46. But why do we call it trisomy? Because it's going to have two of them of the same kind, and then one from the mother. So they become 3. So it becomes trisomy. And because it is happening on chromosome number 21, on chromosome number 21, yes, because it happens on chromosome number 21, therefore we're gonna call it trisomy 21. And remember, tri means 3 and zome means a body, so it's a body made up of 3 kinds of chromosomes of the same kind, and this trisomy is happening in So basically that's how it come to be there. So this is how you explain each nucleus should contain 23 chromosomes after non-disjunction. One cell will contain 22 instead of, and other will contain 24. If the 24 fuses with a normal gamete, it will result in 47 instead of 46, and then this results in Downey's syndrome. Yeah, so you take a screenshot of that's it. So please know how to describe the process of Down syndrome. Then we go to the last question of meiosis, and then we smash genetics. So now they're saying that name the phase of meiosis during which the process represented above takes place. This is crossing over. Automatically is going to be prophase 1. It's going to be prophase 1. They're saying describe the process of crossing over. We explained it, and make sure that you guys, you go when you know how to describe the process of crossing over. We described it properly. So please, I'm not repeating it again. Yeah, but we describe it there. Make sure that you know it, or you take a screenshot of it so that you're able to uh, get the answer correct. And then I say that please don't forget, when it comes to the chromatid, it's supposed to have the word adjacent. Then they're saying that explain the importance of crossing over. We say that the function of crossing over is to bring about genetic variation. And I say that it must be the word genetic. Don't just say variation. It must have the word genetic. Draw the diagram to show the position of alleles, showing this chromatid Y. Don't forget, whenever they ask you to draw, make sure that you know how to draw. Make sure you know how to draw. So in chromatid Y, when after crossing over, they're gonna look like this. It's a box. It's another box. It's another box. So this is X and this is Y, and then this is D. Oh, it's D, and then this is R, then this is capital B. But this one now, the black will join the what? The white, and the white will join the black. It means that I'm gonna have my small b here because the black joined the white one, and then I'm gonna have my black here like this. But I still have my D and R, D and R. Yeah, so that's how it's gonna be. Now you can label, and you can also give the caption. So let's go to the next point, which is genetics. Let's shoot to genetics. Guys, any question from the chat or from the if you have any question, you let me know. Let me know. I don't see any question here from the chat, so it means that we are still moving well. We are still moving well. Okay. Okay. Let's start. The first question is saying that differentiate between dom differentiate between differentiate between recessive and dominant allele. So when you say dominance or dominant allele, this is or dominant character, an allele which expresses itself in heterozygous state is a dominant allele. Recessive allele is an allele which can only express itself in absence of a dominant allele. This one expresses itself, for example, capital F, small f. So a capital F becomes a dominant allele because it can express itself in the what? In a heterozygous state. While this one, recessive, can only express itself in absence of a dominant allele. Phenotype, we say that is the description of an organism in terms of physical appearance, while genetic description of an organism in terms of the genetics is what called the genotype, genetic composition. Complete dominance. Complete dominance is the type of dominance whereby one allele is completely dominant over the other, while incomplete is a type of dominance whereby neither of the allele is completely dominant over the other. So that is it. What is the locus? Is the position of a gene on a chromosome. Then when you talk about Mendel's law of segregation, here you are trying to describe that all organisms have two in monohybrid, two alleles which control a specific character. So these alleles separate during meiosis. You understand? So that each gamete will contain one allele of that particular what? Characteristic. You can't have two of them at the same time. No. You only gonna have one. And basically, they're gonna unite during what? During fertilization. So the genetic disorder which result in the failure of the blood to clot is referred to as hemophilia. We call this one hemophilia. Then we go to the next thing. What is sex-linked character? Is a character whose genes are carried on X chromosome. These are characters whose genes are carried on X chromosome. Then they're saying that what is a sex-linked gene? These are genes which are carried on X chromosome. Give examples of sex-linked disorders or characters. You can talk about hemophilia. You can talk about color blindness. How many alleles control blood group? We say there are three. Allele for A, allele for B, and allele for O. A man of blood group A marries a woman of blood group B. Okay. What is the percentage chance of producing a child of blood group O? Okay. Here you have to go for genetic cross. And you cross and you find out if a man of blood group A marries a woman of blood group B, what is the percentage chance of producing someone with blood group O? So this one, it looks like, let me, genetics is back here. Yes. So it looks like this question. Cabello of blood group A marries Carabo of blood group B, and they produce a child of blood group O. What is the genetic? What is the genetic? Use the genetic codes to show the ratio. You see, here they're asking for the ratio of the offsprings. So you don't forget to start with the P1, F1, P1. Remember, you said P1, F1. So we come and we mark you that. We give you a mark. Phenotype. Phenotype. And then you say genotype. And then you say meiosis. And then you say gametes. And then you say fertilization. So we give you another mark here. Then the phenotype and genotype, you get it from the question. Cabero of blood group A marries Carabo of blood group B. You see, guys? So you tell us that Cabero of blood group A. You understand? Cabello of blood group A. But in this question, the question they asked you, they say that a man, you have a man of blood group A marries a woman of blood group B. So it means that it's going to be a man of blood group A, according to the question now. Blood group A. Don't just say A. A will be wrong. Cross with blood group B. Don't just say B. It will be wrong. Then the genotype, obviously, they say that they produce a child. What's the percentage chance of producing a child of blood group O? Meaning that a man of blood group A. So if both of them they were heterozygous, they were heterozygous, then automatically it's gonna be A heterozygous, and then it's gonna be B heterozygous, and then meiosis takes place, and then you form the gametes. So please, you don't forget these things. Please don't put the A here. Don't modify, don't put it there. This is wrong, this is wrong. And don't put do I like that. This is also wrong. It's supposed to be small I like that. So no stories about it. No stories at all. So we go to the next question. We go to the next question. What is the next question? How many alleles are involved in the dihybrid cross? How many alleles? How many alleles? How many alleles are involved in the dihybrid cross? In this case, we are looking at how many alleles? There are two, because they say dihybrid. Ne? Yes, actually, they're not. How did I say alleles? How many alleles? They're supposed to be four. Mm. Because each in monohybrid there are two, for a capital letter and a small letter, or a capital letter and a capital letter. So in the dihybrid there are four. And how many characteristics are involved here? There are two. If they ask you the characteristics, there are two. But if they ask you the alleles, there are four alleles. So please don't confuse the two. Ne? Yes, they're saying that what is genetic engineering? When you talk about genetic engineering, we are talking about manipulating. Whenever you talk about engineering, you're talking about manipulating. What are you manipulating? You understand? Yes. So whenever you talk about engineering, you are trying to manipulate. What are you manipulating for? Bring about the human needs. So it's the process by which genes or DNA are changed or manipulated to produce different organisms. So you try to manipulate it so that you produce something which are needed. So what are some of the advantages? Basically, it can be used for food. It can be used for research. It can be used, they're asking you the advantages. We say that it can be used for medication, it can be used for pest control, it can be used to increase the food, it can increase the shelf life, but they want only two. And the disadvantages is definitely expensive and then interfering with the nature. Some of them we don't know the potential health impacts, we don't know them. And some of them we don't know the long-term effects of this. You understand? We don't know, we just eat, just enjoy. You don't even read. When you take a bite and it tastes yummy yummy. Yummy yummy in the stomach. Nothing else. Don't wanna know. We will know afterwards. Okay, we go to the next question. What are stem cells? When you talk about stem cells, these are cells which have ability to divide and form other. They're undifferentiated cells and they have ability to divide to form other body structures. So those are what you call the stem cells. What are the sources of stem cells? Here, people are doing using distinction material book. Yes, the bone marrow, the cord blood. This is we're talking about umbilical cord, the tissue, adipose tissue. and then amniotic fluid. These are the examples of what? These are the examples of what? Of stem cells. And then what is it for? What do stem cells use this? Stem cells, they are very important for research. They are very important in replacing the damaged cells, you see. So we can use the research to see how diseases occur without affecting the individual, you see. So stem cells are, at least know two of them. Know two of them. Then we go to the next question. What are sources we are done? What is cloning? When you talk about cloning, we are looking at the process of forming identical organism. You reproduce an organism, yes, which is identical to the parent, exactly the same. So, do we have any advantage or any disadvantage of this cloning process? Yes, they are there, but we will see them when we look at a specific question about cloning. Then they're saying that how many blood group? Why is blood group not conclusive for paternity test? Because there are so many people who share the same type of blood group. What are males? Why are males affected by color blindness than females? Males are affected by color blindness than females because males have one X chromosome. If a recessive allele appears on it, ne, the individual is affected with color blind. Don't say it is affected. Zero is affected with color blindness. While females have two. If a recessive allele appears on X on here, it can be masked by dominant allele on the other one, and the individual is still what? Individual is still normal. So that is it. We need to understand that much more better. All right, let's go to the questions and then we see how we can conclude the genetics. Hemophilia is a sex-linked trait. Hemophilia is a sex-linked trait. Okay? If it's a sex-linked trait, what is happening there? Hemophilia is a sex-linked trait. The pedigree shows the inheritance of hemophilia in the family. So now we have this, we have this, we have that. Ne? Okay. How many generations are those? 3. How many kids are for P and Q? It's 1, it's 2, it's 3. This just got married. Okay. Let's find out what what is represented by a square. A square means a male. How many generations in the pedigree? We said 3. Offspring of P and Q, there are 3. Give the letter of the female who have hemophilia. Remember, it's a circle. So it's 1, it's 2. So it's T and U. So we shall say it's T and U. Give the genotype of R. That's where the story starts. Genotype of R. Genotype of R. genotype of R, genotype of R. It's here. So they say the sex linked. Please don't use anything related to non-sex linked. If you see that this is not sex linked, they have not talked about sex linked, don't use X and Y. So they said sex linked. So I'm gonna say that this is a female. A female can be capital H, small h, or capital H, capital H. But now they have a kid who is male and is sick. It's not sick, it's hemophilic. We don't say sick, or we don't say affected, we don't say sick, it's hemophilic. So if this came from the father, came from here, automatically this came from the mother. You see? So if it came from the mother, then it means that it means that this is X, capital H, Y. While this one is X, because this came from the mother, is gonna be X, capital H. This came from the mother, small h. So it means that R is X, capital H, and then X, small h. So that's what it means. Aha! Then we go to the next question. Give the offspring of the female who have hemophilia. Yeah, so we are done with that question. Guys, okay, let me finish up this. I think when we finish genetics, I'm going to stop here. Yeah, I feel exhausted since morning I've been teaching. All right, so it means that I will stop here, then I will continue the class, I think around 2. So it means that I'm going to have a live which starts from here. Should I stop here? Yeah, I think let me just stop here, guys. I'm going to have a live which is going to start from 2. Let me see from the comment section. Is it fine, guys? Let me know from the comment section. I feel exhausted now. I don't want to be just moving small. I want to be when I'm explaining, and then there is some things which I want to also talk about. So you guys, I think I'm going to stop here. Rendani said, yeah, it's sharp. So that now when I come back, I'll talk about the 4 different types of pedigrees, the 4 different genetic crosses. Ne? So that now tomorrow, what time is best? It can't be okay, that's 2 o'clock. 2 o'clock, guys. I'm gonna put the live reminding you 2 o'clock tomorrow. So tomorrow 2 o'clock, I think I'm gonna come back for the what? For the class. We'll start from here and then finish up with the evolution. Please take note of this. Go back and watch the video. This video I've made 2 o'clock. No, 2 o'clock tomorrow. 2 o'clock. 14. Thank you. Thank you for rectifying that. 14 hours. 14 hours. Yes, that's when you're gonna be starting. 14 hours. So that I'm able. Yes. 14 hours tomorrow. So try to go through what I've done. Rewatch it. Make sure that whatever I've talked about, you know them. So that by the time we talk about at 2 o'clock, we will make it from 2 o'clock until 4 o'clock. Then the class will be done. Tomorrow, 14 hours. When I say 2 o'clock p.m., 14 hours. Ne? 2, 16 hours. Tomorrow on Sunday. So that, yes. Okay. I'll be here. Let me also set the alarm. All right, guys, please make sure that you try to sleep so that tomorrow is wire wire. Wire wire means that we have to push until we see that distinction is there. Yes, I don't want to break down. Ne? Yes, I don't want to break down. So I still have a lot for you. You see, I have to talk about all this. I have to talk about all this. I have to talk about all this. Yeah, we're almost there, but yes, actually, it's half of it. So thank you so much, guys, who have been on live. Amesi. Yes, thank you so much, guys. I think I'm going to stop here. Please go back and read tomorrow, 2 o'clock. We meet again. All right, 2 o'clock. I will set the live, then I'll try to share with you guys. Thank you so much. See you again in Paul. I see you. Paul, Amesi, Junior, Rendani. Yes, Vilakazi. All right.